Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue our review, getting ready for the state test today. Today, this is the standard that we're looking at, and I'll go ahead and put those standards up here so we can talk about them in a little bit more detail. So today, we're really going to be talking about the cell membrane. We're going to talk about how it helps to maintain homeostasis. We're going to talk about those different types of transports, and we're going to talk about a few different solutions that cells can be in. You might remember the words hypertonic, hypotonic, and isotonic, and so we're going to talk about all of that stuff today and give you all a little bit of a refresher before you have to take your state test at the end of the year. So let's go ahead and get the ball rolling on this. So let's talk about the cell membrane. So the cell membrane is an organelle. It's the boundary of the cell. The cell membrane is responsible for controlling what comes into and out of the cell. We say that this is selectively permeable. To be permeable means that something can flow through it, like a rock with a lot of holes might be permeable. Because the cell membrane can choose what comes in and out of it, it is selectively permeable. Some things can enter and leave, but not everything can. By controlling what comes into and out of the cell, the membrane helps maintain homeostasis. Remember, homeostasis is the maintenance of a stable internal environment. We don't want just anything coming into the cell because toxins and poisons could disrupt the cell, but we do need things like nutrients and water. And so some things can come in freely and other things can't. Okay? So the cell membrane, how does the cell membrane help to maintain homeostasis? Well, there's really two ways that it can control what comes in and out of the cell. We can do something called active transport, which is when we're moving things into or out of the cell by using energy. And again, that energy is in the form of ATP. We can also do something called passive transport, which means moving things in or out of the cell by using the natural flow of molecules down the concentration gradient. Remember that a concentration gradient means how compact are things in one area. Things tend to have a natural tendency to want to spread out, right? Imagine you are walking onto the bus in the morning and you see that the front 10 seats are all filled up with somebody and the back 10 seats are all empty. Would you go and sit by somebody that already had somebody sitting on that seat, or would you naturally want to spread out a little bit? The same thing's kind of going on with these molecules. They're not trying to do it, it's just a natural tendency. Again, this passive transport, this is maybe you've walked into your house and your mom is cooking food and you can smell what she's cooking at the front door. Well, that's because those molecules are spreading out from the kitchen and just kind of filling up that whole space, right? So we're going to go in and talk about these different active and passive transport and some different examples of each one here next. So let's talk about active transport first, okay? Active transport is moving things against the concentration gradient, which requires energy in the form of ATP. There are a couple of different types of active transport. So you can have something called endocytosis, which means we're using energy to bring things into the cell. Let's just look at the word endocytosis. So first of all, that prefix endo means inside, and then cyto means cell. So if we're bringing something inside of the cell, it's endocytosis, okay? There's two types of endocytosis. We can have something called penocytosis, which is when we're bringing liquids into the cell, or we can have something called phagocytosis, which is when we're bringing solids into the cell. A good example of phagocytosis is, imagine an amoeba eating something. When an amoeba eats something, it will completely surround it and then bring it into the amoeba. And so that is a form of phagocytosis. Okay? The opposite of endocytosis is exocytosis. In exocytosis, we use energy to move things out of the cell. This might be us trying to get rid of some waste or something like that, and so we would use energy to make it leave the cell, and that's called exocytosis. To me, that kind of sounds like the word exit, and so that's just an easy way to remember that exo is moving it out and endo is moving it in. Okay, great. So let's look at some different examples of passive transport. In passive transport, this does not require energy. Molecules move down the concentration gradient. Okay, there's a couple different types of passive transport. First of all, we can have simple diffusion. This is the natural tendency of molecules to want to spread out. Like I was saying earlier, Imagine you're upstairs in your room and then all of a sudden you smell the scent of freshly baked cookies, right? The cookies aren't in your room yet, they're down in the kitchen, but the molecules that you're smelling are spreading out throughout the house. That's called simple diffusion. Things just naturally spread out from each other, okay? Another type of passive transport rather is called facilitated diffusion. This is simple diffusion of larger molecules. If a molecule is too large, it will need a little help from a channel protein. So what facilitated means is help. If you facilitate somebody learning something, you help them learn, right? In facilitated diffusion, this is still moving down the concentration gradient, just like simple diffusion, but usually this is with large molecules, and the molecule is too big to go through the cell membrane on its own. So instead it will use a channel protein, okay? And then lastly, another type of passive transport is osmosis. Osmosis is the movement of water from low to high solute. And we're going to talk about osmosis in a good bit of detail here on the next couple of slides. So let's talk about osmosis. What I really want you to remember is that if water is moving instead of solutes, then it is considered osmosis. So if water is the thing that's moving across the membrane, we would consider that osmosis. There's a couple of different types of solutions that you need to know about. Really just try to remember this sentence. Water always moves towards high solute. Water always moves towards high solute. If you can remember that, that's going to make all of this stuff a little bit more easy to remember. Okay, so let's talk about a hypertonic solution. Okay, in a hypertonic solution, this means that the solution outside of the cell has more solutes than the cell itself, and this means that the cell will lose water and shrink. And an example of this is plasmolysis. If you put a plant, sometimes what will happen, imagine you have the road's ice over, right? So imagine it's really cold, the road's ice over. What will often happen is people will lay down a bunch of salt so that the ice melts quicker, right? But what will happen is all of that salt then goes into the median and the plants on the outside of the road, and those plants will start to lose all of their water because now there's a whole bunch of salt in their environment. What will happen then is because those plants are losing all that water, they'll start to wilt and they could potentially die. That's called plasmolysis, okay? Another type of solution is called isotonic. And an isotonic solution outside of the cell has the same amount of solutes as the cell. The cell here will stay the same size because the concentration of solutes is the same. The last type of solution you need to know about is called a hypotonic solution. This is the opposite of a hypertonic solution. In a hypotonic solution, the solution outside of the cell has less solutes, and this will make the cell gain water and grow. It could potentially grow so much that it ruptures, and if it ruptures, it's called cytolysis, and that's very bad because now the cells are destroyed. Okay? So sometimes people see this hypotonic solution, they see that the cell will grow, and a little mnemonic device for that is that it will swell up like a hippo. So hypo. That's kind of what's going on there. Just try to remember what these prefixes mean. So hyper means over and above. So in a hypertonic solutions, there's more solutes outside. In a hypotonic solution, hypo means below, like hypothermia. You've probably heard of that before, when your temperature is too low. In a hypotonic solution, what's going on here is that there's less solutes outside of the cell, and so it's going to swell up like a hippo. Okay, so y'all, that's just some stuff I wanted to go over with y'all. Let's just take a look at the pictures, and then we'll kind of wrap up for the day. Okay, so again, in osmosis, remember that osmosis is when water moves. If the water is moving, it is osmosis. Let's look at these three different types of solutions. So these are all red blood cells, okay? If your red blood cells are in a hypertonic solution, hyper means that there's more solutes outside of the cell. Remember that water always moves towards high solute. And so that means that water is going to be leaving the cell, and if that happens, they're going to start to shrink, and that's not going to be good, okay? The next type of solution is called an isotonic solution. Iso means equal, so there's an equal amount of solutes inside and out of the cell. Here, because it's equal, there's some water moving out and there's some water moving in, but it's in equal amounts, and so the cell stays the same size. Okay? The last one is called a hypotonic solution. Again, hypo means lower or low, and so in this there's more solutes inside of the cell, there's less solutes outside of the cell. That means that a lot of the water is going to move into the cell, and the cells are going to swell up, and they might even get so big that they rupture, and that's called cytolysis, and now this cell is destroyed and it's not usable. So y'all, thank you so much for making it here today. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.